Assalamu alaikum dear students. Today we will discuss PCR Polymerase Chain Reaction MCQs. The template for PCR is option A RNA, B single stranded DNA, option C double stranded DNA, and option D none of these. So the correct option is C double stranded DNA. PCR consists of option A 2 steps, B 3 steps, C 4 steps, and option D 5 steps. So the correct option is B 3 steps. RNA populations can also be used as PCR templates after reverse transcription into option A cDNA, B tRNA, C mDNA, and option D rRNA. So the correct option is A cDNA. Subscribe now and press the bell icon. Never miss an update from a technologist. The efficiency of amplification in PCR in later cycles is reduced due to option A reduction in substrate concentration, B insufficient enzyme and time to synthesize a mass quantity of DNA, option C buildup of PCR product which competes with primer for hybrid formation, and option D all of these. So the correct option is D all of these. Which of the following infections can be diagnosed using the polymerase chain reaction? Option A HIV-1 and HIV-2 viruses, option B hepatitis B viruses, option C Mycobacterium tuberculosis, option D all of these. Correct option is D all of these. The original enzyme used in the PCR reaction is known as option A E. coli DNA polymerase, B E. coli RNA polymerase, C TEC polymerase, and option D all of these. So the correct option is A E. coli DNA polymerase. The cloning step in PCR sequencing analysis of microbial communities is necessary for option A the amplification process, B preventing contamination by outside DNA, C separating the different rRNA gene sequences in the mixture, and option D none of these. So the correct option is C separating the different rRNA gene sequences in the mixture. The viruses that can achieve neoplastic transformation are called option A DNA tumor viruses, B RNA tumor viruses, C retroviruses HIV, and option D none of these. So the correct option is A DNA tumor viruses. Double stranded DNA denaturation with a specified limit of temperature is option A reversible reaction, B irreversible reaction, C either A or B, and option D none of these. So the correct option is A reversible reaction. For gene transfer to be effective, transforming DNA must be option A incorporated into the bacterial chromosome, B incorporated into a viral genome, C free in the bacterial cytoplasm, and option D none of the above. Correct option is A incorporated into bacterial chromosome. genomic libraries are made from option A. Genomic DNA of an organism, B. Genomic RNA of an organism, C. Genomic cDNA of an organism, and option D. Genomic mRNA of an organism. So the correct option is A. Genomic DNA of an organism. From a single molecule of DNA, PCR can make option A. One additional copy, B. Hundreds of copies, C. Thousands of copies, option D. Millions of copies. So the correct option is D. Million of copies. PCR is used option A. To diagnose genetic disorder, option B. To solve crimes, C. To study gene function, and option D. All of these. Correct option is D. All of these. Bacteriophages are option A. Cells in the blood that eat bacteria, B. A class of bacteria, C. Bacterial viruses, option D. None of these. So the correct option is C. Bacterial viruses. Specialized transduction occurs when option A. The bacteriophage incorporates randomly into the bacterial chromosome, B. The bacteriophage never incorporates into the bacterial chromosome, option C. The bacteriophage always incorporates at the same position in the bacterial chromosome. So the correct option is C. The bacteriophage always incorporates at the same position in the bacterial chromosome. PCR can be used to amplify a specific fragment of DNA. Which of the following can be used? Option A. A drop of blood, B. A hair follicle, C. A fragment of skin, and option D. All of these. So the correct option is D. All of these. To clone into a plasmid vector, both the plasmid and the foreign DNA are cut. Option A. With the same restriction enzyme and mixed, B. With different restriction enzymes and mixed, C. With the combination of enzyme and then separated, option D. With the combination of enzyme and mixed. So the correct option is A. With the same restriction enzyme and mixed. Transformation means option A. Formation of a pilus, B. Acquiring DNA from the bacterial cell environment, C. Plasmid containing a F factor. Correct option is B. Acquiring DNA from the bacterial cell environment. Restriction maps. Option A. Allow comparison between DNA molecules without the need to determine nucleotide sequence. B. Allow comparison between DNA molecules but requires determining the nucleotide sequence. Option C. Does not allow comparison between DNA molecules. So the correct option is A. Allows comparison between DNA molecules without the need to determine nucleotide sequence. Which of the following is correct? Option A. HIV is a retrovirus that kills human helper T cells. Option B. Causes acquired immune deficiency syndrome. C. Destroys the immune systems. And option D. All of the above. So the correct option is D. All of the above. PCR is an abbreviation for option A. Protein change reaction, B. polymerase chain reaction, C. Positive charge reaction, and option D. Preliminary change rate. So the correct option is B. Polymerase chain reaction. A PCR cycle consists of option A. 3 steps: denaturation, primary annealing, and elongation. Option B. 3 steps: denaturation, initiation, and elongation. And option C. 3 steps: primary annealing, elongation, and termination. Correct option is A. Denaturation, primary annealing, and elongation. PCR can make multiple copies of option A. DNA, B. RNA, C. protein, and option D. carbohydrates. Correct option is A. DNA. Southern blotting involves option A. electrophoresis of DNA molecules and then blotting the separated DNA bands followed by incubation. B. electrophoresis of DNA molecules and then blotting the separating RNA bands followed by incubation. Option C. electrophoresis of DNA molecules followed by incubation. So the correct option is A. electrophoresis of DNA molecules and then blotting the separated DNA bands followed by incubation. PCR can be used in option A. cloning, B. sequencing, C. medical diagnosis and forensic medicine, and option D. all of the above. So the correct option is D. all of the above. PCR amplifies millions of copies of option A. short fragment of DNA, B. the entire genome, all the DNA in a sample, C. both A and B, and option D. short fragment of RNA. So the correct option is A. a short fragment of DNA. Additional monosaccharides are added in the Golgi to produce option A. either a high mannose type oligosaccharide or a complex type, B. either a fructose type monosaccharide or a complex type, C. either a high sucrose type polysaccharide or a complex type. So the correct option is A. either a high mannose type oligosaccharide or a complex type. Bacteriophages bind to a bacterial surface and inject the phage DNA through the option A. cell wall into cytosol, B. cell wall into plasma membrane, C. both A and B, and option D. cell wall into the endoplasmic reticulum. So the correct option is A. cell wall into cytosol. In a PCR reaction, after 4 cycles, each molecule of duplex DNA will give rise to option A. 16 single stranded of DNA, B. 16 double strands of DNA, option C. 18 single strands of DNA, and option D. 18 double stranded DNA. So the correct option is A. 16 single strands of DNA. The PCR polymerase chain reaction is becoming the method of choice for option A. alteration of gene, B. screening gene, C. sterilization of the gene, and option D. all of these. So correct option is B. screening gene. What technique can be used to determine the Murder who left blood with the victim. Option A, DNA sequencing. B, PCR amplification. C, Western blot. And option D, RFLP mapping. Correct option is B, PCR amplification. Northern blotting is option A, widely different than southern blotting. B, another name for southern blotting. C, analogous to southern blotting. Correct option is C, analogous to southern blotting. Thank you for watching my videos.